Hi students, this is A. Mandri Kumar Rajesh. In this video, we are going to discuss a microorganism called bacteria. How can we define bacteria? Bacteria can be defined as unicellular prokaryotic cosmopolitan microorganisms. Unicellular prokaryotic, then cosmopolitan microorganism. So you may know about these two words, unicellular and prokaryotic. Then what is called cosmopolitan? Cosmopolitan means that bacteria, that microorganism can found all over the world. So bacteria can found all over the world. Then the branch which deals with the study of bacteria is called as bacteriology. So the branch which deals with study of bacteria is called as bacteriology. Then the bacteria was first discovered by Dutch scientist Anton van Leeuwenhoek in the year 1676. In the year 1676, and he considered and he named the bacteria as animalcules. He named the bacteria as animalcules. Animalcules means little animals. Animal and cules means little. Little animals are microscopic animals. So Anton van Leeuwenhoek considered bacteria are little animals or microscopic animals. Then whether the bacteria are friend or foe. Whether the bacteria are friend or foe to us. I give you some examples. So it can act like a friend in some condition. For example, in our digestive system, in our large intestine, a bacteria called E. coli is present. So a bacteria called E. coli is present in our large intestine. So that E. coli can break down the food materials and it can absorb nutrition from the food materials, and it can assist to produce vitamin K. So this E. coli, which is present in the large intestine, it can assist to produce vitamin K. So vitamin K is important for blood clotting. So vitamin K is important factor for blood clotting. So E. coli is a bacteria which is present in our large intestine. It can break down the food material and it can absorb the nutrition, and it can assist to produce vitamin K. That vitamin K is important for blood clotting. Then another example. In our day-to-day life, we used to consume curd mixed with the rice. We can use that curd. So how this curd is prepared? In our home, our mom used to add some amount of fermented buttermilk which contains the microorganism Lactobacillus aceti. So our mom add that fermented buttermilk to normal fresh milk. So in course of time, the normal fresh milk converted into curd by the activities of Lactobacillus aceti which are present in the fermented buttermilk. So that Lactobacillus aceti act on lactose and it converts lactose into lactic acid, and that milk converted into curd. So in this condition also, the bacteria act like a friend. While in some cases, in some condition, the bacteria are act like foes. That means enemy. It can cause harmful activities. So typhoid. So it is a serious disease which is caused by the bacteria Salmonella typhi. And another example, tuberculosis. It is a very dangerous disease which is caused by the bacteria Mycobacterium tuberculosis. So in these two conditions, the bacteria act like force. It gives harmful effects. Suppose if a bacteria which gives beneficial effect, that bacteria are called as probiotics. Whether the bacteria gives the harmful effect, the bacteria are called as harmful bacteria. Then I am going to tell about milestones in bacteriology. Milestones in bacteriology. Let me tell you the milestones in bacteriology. In 1829, C.G. Ehrenberg coined the term bacterium. So, who coined the term bacteria? C.G. Ehrenberg. Until then, the bacteria are considered as animalcules. So, in 1829, C.G. Ehrenberg coined the term bacteria. Then, in 1884, Christian Graham introduced Graham staining method. So with this staining method, we can differentiate bacteria into two types. They are gram-positive bacteria and gram-negative bacteria. If the bacteria retains the purple color, they are considered as gram-positive. If the bacteria do not retains the purple color, they are called as gram-negative. So the gram-negative bacteria are very dangerous. They can cause serious diseases. Then in 1923, David H. Berge published first edition of Berge's manual. So this manual contains enormous information about the bacteria. So that manual was published by David H. Berge. Then in 1928, Frederick Griffith discovered bacterial transformation. It is a type of sexual reproduction. So the bacteria can transfer their genetic material between the bacterium. So that sexual reproduction was discovered by Frederick Griffith. Then in 1952, Joshua Ledberg discovered of plasmid. Plasmid is nothing but the extra chromosomal material which is present in bacteria. So in bacteria, it contains circular DNA. In addition, it contains plasmid. That means extra chromosomal material. So this plasmid is a good vector in biotechnology. In biotechnology, these plasmids are used as vectors. So that's all about the milestones in bacteria. Now I am going to tell about the salient features of bacteria. So please wait a minute. Let me teach you the characteristic features of bacteria. Here I have written some of the general characteristic features of bacteria. They are prokaryotic organisms and lack nuclear membrane and membrane-bound organelles. That means the bacteria do not possess well-defined nucleus. They contain primitive nucleus and they lack nuclear membrane. Then the membrane-bound organelles. So they lack membrane-bound organelles like mitochondria, endoplasmic reticulum and Golgi apparatus. And bacteria, the nucleuses are naked because nuclear membrane is absent. That is why that nucleus is called as naked nucleus. Then the genetic material is called as nucleoid or genopore or incipient nucleus. So nucleoid means it is a nucleus-like structure. So it is a chromosome which do not possess nuclear membrane. So nuclear means it is a nucleus-like structure. Then genopore means it refers to a prokaryotic chromosome. Genopore means it refers to the prokaryotic chromosome which contains nucleic acid. So that nucleic acid may be DNA or RNA, and the genopore do not contain histone proteins. So it contains nucleic acid but it do not possess histone protein, and that structure is called as genopore. Then incipient nucleus. Incipient nucleus means they are immature nucleus. They are immature nucleus and they have additional structure of chromosome that is called plasmids. I already told you plasmids are otherwise called as extra chromosomal material. So in addition to circular DNA, it contains extra chromosomal material called plasmid. Then the cell wall is made up of polysaccharides and proteins. That means the cell wall is made up of peptidoglycan and mucopeptide protein. The cell wall is made up of peptidoglycan and mucopeptide protein. Then most of them lack chlorophyll, hence they are heterotrophic in nature. Example, Vibrio cholerae. So most of the bacteria do not have chlorophyll, so they do not prepare their own food because of the absence of chlorophyll. So they are heterotrophic in nature. Example, Vibrio cholerae. But some are autotrophs and possess bacteriochlorophyll. Example, Chromatium. So in blue-green algae, nowadays blue-green algae are considered as cyanobacteria. So blue-green algae contains chlorophyll. They can do photosynthesis and they can prepare their own food. So all the blue-green algae are example for autotrophs. Then they reproduce vegetatively by binary fission. So a single cell get constricted and it divided into two cells. So that is called binary fission. And endospore formation. During unfavorable condition, they can produce a endospore, that is cyst formation around the bacteria, and that condition is called as endospore formation. Then they exhibit variation which are due to genetic recombination and is achieved through conjugation, transformation and translation. And these three processes are very important process, and these three processes are considered as sexual reproduction. So during the sexual reproduction only we can get variation in the organisms. So here they tell us they exhibit variation which are due to genetic recombination and is achieved through conjugation, transformation and transduction. So sexual reproduction takes place in bacteria. They are conjugation, transformation and transduction. So this process leads to variation in the bacteria. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.